Hello and welcome to our PowerPoint here on the key knowledge points 7 and 8 around species relatedness. Specifically, you can see here we're going to be looking at the evidence of relatedness between species around structural morphology, including homologous and vestigial structures, and molecular homology around DNA and amino acid sequences. We'll also look at the use and interpretation of phylogenetic trees as evidence for the relatedness between species. So when we talk about relatedness between species, we're looking at all the different species that exist in the world and trying to understand how they're connected. And there's a range of different pieces of evidence that scientists will turn to help them work this out. We've already looked at the first one, their paleontology, in the KK5 notes, and that's specifically around fossils. And we can also look at biogeography, developmental biology, comparative anatomy, and molecular homology. And all of these feed into our understanding of the relatedness between species. So biogeography, just more of an aside here, because VCAR doesn't require you to remember this as a type of evidence here, but it's worth knowing in the back of your head anyway. It's this idea that we can look at how life forms are distributed around the globe, and we're looking at their geographic locations, and specifically the locations of ones that we know are related to each other. You can see there in the diagram, we're looking at modern members of the camel family in terms of the dromedary camel, the Bactrian camel, and the llama. They all are part of that camel family, but you can see that they live in very different areas around the world. And so we can look at where they live to help us get an understanding of where they may have come from, how things have spread out over time, and those sorts of things. So they can help us with that understanding of how they evolved, when did they evolve, where did they spread from as their point of origin. And really what we're doing is we're looking for related species in different geographic areas, or if you don't have living members of a species, you might be looking for the fossils of related species in different geographic areas to get an idea where different organisms lived at different times and how they got there. The next one is developmental biology. Again, this is one that VCAR doesn't require you to remember, but it's worthwhile having this in the back of your head as a type of evidence for species relatedness. Now, often when we think of developmental biology, we think very much of Haeckel's drawings of the embryos at different stages in development of the different organisms and how they look similar at different stages. It's worth keeping in mind that there was a bit of artistic license around that diagram, so it's maybe not the best thing to refer back to. And developmental biology nowadays is a much broader topic where we're particularly looking at things like what genes are involved at what point in development and what do they lead to the production of in that organism. The diagram here is looking at Hox genes, HOX genes, and these are genes that are found in a lot of different animals like insects and other vertebrates, and they're helpful in controlling embryonic development. And depending on when those genes are activated, how long they're activated for, and in conjunction with what other genes they're activated with, you can see them causing the development of different structures that leads to different organisms. Both Mus muscalis, which is mice, and Homo sapiens, which is us, have these genes and have them being activated, but you can see that they're not necessarily activated all at the same time and in the same way as they would be for, say, the fruit fly Drosophila melanogaster. So there's some differences there, and that's what we can do nowadays because we have a better understanding of the things that control development. And so we're really looking at the similarities and the differences in how organisms develop, and the theory is that the more similar the patterns of development, the more closely related those two species would be. There's more that particular focus on genetic basis of phenotypic structures nowadays than just what you can see, what's the actual genetic basis for it, how those features change during evolution, and how novel or new structures arise. So that's developmental biology. Here we have exam question 1. It says biogeography is the study of A, the distribution of plants and animals in particular areas, B, fossil types and their relation to living organisms, C, islands and their distance from continents, or D, mountain ranges and rivers. So you can pause the video, pick your answer, and then check it against the video in the description below. So both biogeography and developmental biology are things that VCAR hasn't set out for you to know. What they have asked that you are familiar with is certain elements of comparative anatomy. Now, comparative anatomy is, unsurprisingly, where we compare the anatomy of different organisms. The other word here you might hear, or the term, is structural morphology, where you're looking at the similarities or studying the shape of different structures in different organisms. But it's not just necessarily about the shape, it's also about the function of those different anatomical structures. And so we're looking at structures across different organisms to see where do they share structural features, or where maybe do they share no structural features, but rather they share function. And so depending on what's similar and what's different, we can group them into two different categories of structures: homologous features or analogous features. Homologous features are ones where the shape is similar and the structure is similar, but they do different jobs. They have a different function. Analogous is the opposite. They have a different shape, but they do a similar function. So these are our two main categories. And you can see some of those highlighted in the diagram on the right there. You may also find that some organisms have vestigial structures. These are structures in an organism that are reduced or non-functional versions of a feature, and they're there, but they don't serve any purpose. They're little to no present purpose in that organism. Even though they don't currently do anything, they might help indicate shared evolutionary ancestry. Where we look at it and go, well, hang on, this organism has this structure that doesn't currently do anything for it, so where did that structure come from? And that can be sort of traced back to help indicate, oh, here's a common ancestor where that was in use. And that can help us work out how things have evolved over time. So a little more detail on homologous features. Homologous features we particularly connect in with what we would call divergent evolution, where there is a common ancestor, and over time populations of that common ancestor have been put into different conditions, particularly if we're thinking allopatric speciation, where different geographical features may have separated groups and put them in under different selective pressures. And over time, different things have been selected for, and so we've seen changes in these groups into different species over time. And here we're particularly looking at an example with the pentadactyl limb, where that's gone so far that we're talking very, very different organisms, but they have retained a structure that is consistent across all of them. Not perfectly identical, but it contains a similar structure of bones across all these different organisms. That structure is known as a pentadactyl limb. It's a limb that ends in 5 digits. A little bit of variance there. I'm not saying that a horse's hoof necessarily has 5 digits, but it is still considered a pentadactyl limb, or to have arisen from a pentadactyl limb. And so what we can see here is a consistent base structure across all these organisms. And so you've got humans and horses and whales and turtles and frogs and birds, and they all have this variant pentadactyl limb. They have some variant of it. And it's the bone arrangement in particular. You'll notice that the diagram has it color-coded. And so the underlying structure is the same. That's where the homo part of homologous comes from. That's because they have the same structure. Now, in these examples, they actually do very different things. Human hands are adapted for tool manipulation. We have a power grip and a precision grip, whether it's holding something precisely or with a lot of strength. Bird and bat wings are adapted to fly. Horse hooves are adapted for galloping. Whale and dolphin fins are adapted for swimming. Turtles obviously would be for swimming as well, maybe a bit of walking. Frogs for leaping off and pushing off things. So there's a range of different functions that these limbs are used for across these different animals now, but their fundamental structure is consistent, and that's why they're considered homologous features. Analogous features, on the other hand, are the opposite. We might look at these structures and they have completely different underlying structures, but they do the same job. And this is where we would be thinking about convergent evolution, where we start off with two distinct ancestral species, but because they get put into similar environmental conditions, over time, the same sorts of features are favoured, and therefore those sorts of features are being selected for. Even though underlying them, the structure can be different, you can still end up making it the same way. You can see here that we have an insect and a bird. Both of these can fly, and so they both have wings to allow them to fly. However, the way those wings are composed are completely different. Another one to think of might be between sharks and dolphins. They have very similar features. They're very streamlined in the body. They have dorsal fins. They have flippers. They have tails that have a similar shape, even though they move in different directions. And that's going to come down to the fact that they live in the same environment. So water is going to put pressure on those that are more streamlined and favour that. Those that are less streamlined are going to find it harder to survive. And the dorsal fins and the flippers will help movement and stability through the water. And so those are favoured traits that we see being accumulated in two different organisms. So analogous features are those with different structures, but they're doing the same job. So our vestigial features are where we've had an organism evolve, but there's been some feature that no longer doesn't really do a function, but it's not necessarily causing any harm to the organism. So there's nothing to select against it to cause it to be unfavored. It just doesn't do anything. It's not particularly favored anymore. So as these structures don't harm the organism, they're never selected against and they're never properly lost out of it. They're not having nature sort of work against them to get rid of them. And because of that, so they're considered vestigial structures, and we can look at them to help understand where an organism maybe has evolved from. Some examples would include the pelvic bone in whales. You only need a pelvis if you've got legs and are walking on land, so why would whales have a pelvis? And they do. They have a very rudimentary pelvis bone. It's not as developed as it would need to be if they were actually going to walk on land. But it does seem to indicate that they might have had an ancestor who did walk on land. And that's where it's come from. When they're in the water, it doesn't help them, but it also doesn't hurt them in any way. So there's nothing trying to actively get rid of this trait. In humans, we have a coccyx, which is also known as your tailbone. It's a little bone that's an extension of the spine. And really, this is what we see in organisms that have a tail. You'd see more bones along from the coccyx out, and that would lead into the tail of the organism. But we obviously don't have tails. And so this is not something that is needed in humans, but it might indicate that we had some ancestor with a tail. That being said, it is possible we tend not to see people really being born with a tail. The tailbone or the coccyx is internal to their body, but it is entirely possible at times for people to be born with what we would call a vestigial tail, where there is an overgrowth of that bone outside the body, and a lot of the time people have that surgically removed nowadays. You may have also heard that the appendix is considered a vestigial organ, and it was for a long time until in the mid-2010s when it was actually discovered that it played a role in the immune system, and so now we know that it's actually not vestigial because it does have an active function and is useful to humans today. So that's why the appendix is no longer considered a vestigial structure.
Thanks for stopping by. Today's video is a follow-up video from last week's homologous and analogous structures, and this week we're talking about vestigial structures. Now, vestigial structures are left over from evolution. They're a structure that used to have a function in an ancestor species, but is useless now in the modern species. So the first example we're going to look at is in humans, and that's the 𝗃𝕃𝕃𝕃 muscle. And the 𝗃𝕃𝕃𝕃 muscle is responsible for giving you goosebumps when you get the chills, or when you have an adrenal response because you're feeling anxious or scared about something, and you get goosebumps. And animals now, they still do this, but their fur puffs out. You look at bears and monkeys and things like that, and they do it either when they're cold to stay warm or when they feel a threat to make themselves look bigger. But for us, it doesn't work that way, right? We don't have enough hair to make it have an effect, so it's really useless for us. But anciently, if you look back at our ancestors, they used it for the same purpose that many animals do today. The next thing we're going to look at is the coccyx, or the tailbone. Again, in humans, it's not functional at all. It serves no purpose. If you went without it, you'd be totally fine, but it's there all the same. And again, it shows a structure left over from our ancestors which had tails. And then the last one I was going to talk about is the baleen well. The baleen well has this remaining hip joint here, which is reminiscent of its ancestors, which had forelimbs. Of course, the whale has no forelimb or anything there, but it still has those bones from the hip joint.
So moving now from physical structures, we're going to look at molecules. And what we would term this area of study is molecular homology. We're looking for molecules that are the same and that have the same structure themselves. That's where the homo or homology part of this comes in. So all living things have some commonality in their biochemistry, and that indicates some sort of common ancestor that everything is descended from. And one thing we can look to that's most basic here to give us this idea is the fact that we share the basic DNA structure with every other living thing on the planet. Now, we can go from that though, and there are obvious differences in what those sequences are of that DNA. And so those similarities and differences that we can see in the actual sequence, those are going to help us work out how related two organisms are. The more matched those DNA sequences are, the more closely related those two organisms are going to be, because there's been less time for mutations and differences to accumulate. Whereas on the other hand, if you have less matches, that indicates a further or more distant relationship, and that the branching before the common ancestor for those two species was much longer ago. There are two main techniques that we use to investigate how similar organisms' biochemistry is: DNA hybridization and amino acid sequencing. These aren't the only two, these are just a couple of key techniques to be familiar with. So a little bit of a rundown about DNA hybridization, and this is actually illustrated in the diagram on the right there. DNA hybridization is where you take the DNA from two species for a sequence of DNA that is comparable between the two. It should be like the same gene, or at least as similar as you can get to it being the same gene. And you're going to take those two sections of DNA and you're going to separate the two strands in each section, and then you're going to take one from one species and one from another. You're going to mix them together and you're going to see if you can get them to join up. Now, if these species are closely related, you would expect that most of that sequence is going to be the same, and you'll get a lot of bonding and a lot of matching between them. If, on the other hand, they are not closely related, you would expect more differences to be present, and therefore you're going to have more spots where those two sequences will not match up. Once you've done this, you now need to work out how much has matched up and how much has not matched up. And this is a bit of a sort of series of logical steps you've got to work through here. And the idea is that the more matches there are, the more bonds that have been made. The more bonds that have been made, the more energy it would take to separate those bonds. Therefore, if we apply heat energy to the DNA and we wait to see when those bonds are broken, the one that has more bonds will take a lot more energy, therefore a higher temperature, before it actually breaks. So what will happen with DNA hybridization is you've separated the two original strands into two single strands, try to combine them, and now you've got this left over, and you're going to try and see how long it takes before those two strands of the two different species that you join together, how long does it take before they actually separate? And so you heat it up, and you will record the melting temperature, and that's the point at which the strands have separated. If that's a really high temperature, you know there were lots of bonds that it had to break, and you need a lot of energy to do that. And if there's a lot of bonds, that means there's a lot of matches, that means they're really closely related. On the other hand, if you didn't need as much energy and it's a lower temperature, that would indicate you had less bonds to break, and therefore it is a more distant relationship. And so you can use that to work out how closely related things are. It's not as precise necessarily as looking at the individual sequences and doing actual DNA sequencing, but if you're not able to do that, this might be a good alternative. The other technique that you could use is, instead of looking at the DNA itself, look at what the DNA makes, and that is amino acids in the proteins that the DNA is leading to. And so instead of sequencing DNA, you might sequence a protein to get its list of amino acids in order. You can then compare those between two species, and you can see where those sequences of amino acids for a protein that's common to two species, where that may be similar and where it may be different. Again, the more similarities, the more related those two species would be, and the more differences, the less related they would be. On a side note, you've probably heard of mitochondrial DNA. Most of the time when we talk about DNA, we're talking about what we would call nuclear DNA, DNA we find in the nucleus, though there is actually a little bit of mitochondrial DNA, and plants actually have a little bit of chloroplast DNA as well, but obviously we're not plants. So with mitochondrial DNA, we're interested in it because it can also be used to determine relatedness. But it's a very specific way it's used, because it's believed that mitochondrial DNA is only passed on via the maternal line. That means through your mother's 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 line, basically. So you will have the same mitochondrial DNA as your mum. She will have the same as her mum, so you actually have the same as your mum's mum, who will have the same as her mum, who will have the same as her mum, and so on and so forth, all the way back in time. Obviously, the further back in time we go, the more chance that some mutation has occurred at some point, and there might be a difference that's accumulated. So here we have exam question 2. You have the same mitochondrial DNA as your A, paternal grandfather, B, paternal grandmother, C, maternal grandfather, or D, maternal grandmother. You can pause the video if you'd like and pick your answer, and then check it against the video linked in the description box below. So let's talk about how we might compare DNA. There's a couple of things we might do here. Obviously, we can do DNA hybridization, and that can be great for giving us a percentage of similarity between two strands, but we might also want to do a direct comparison of DNA-based sequences and actually look at the individual bases themselves. And you might do that just for a certain section, or you might decide to do a comparison of the entire genome between different species and look at the similarities and differences between all their genetic material. If you are going to compare DNA-based sequences, it's really important that you pick similar, if not the same, genes to do that with. If you're picking different genes, of course, you're going to get lots of differences. It's going to invalidate your results. And if evolution has occurred, when we look at our results, we can work out and predict who is most closely related to who based on the similarities and differences. The more similarities, the more closely related they are going to be. So at the bottom there, you've got part of the hemoglobin gene, and you've got a sequence of that. And you'll see from what you've got there that in red underneath humans all in black, but for orangutan, monkey, and rabbit, every time there is a different letter, it's in red. And so you can see a few differences there between humans and orangutans, humans and monkeys, humans and rabbits, and all of that. And those would be places where you'd be thinking, okay, there's been a substitution here. The base letter has been swapped for something else. So that might be what led to that. Then you'll notice that there are some, with the human and orangutan have a dash, and then underneath the monkey and the rabbit both have 2 red A's. And in this case, one of two things might have occurred. Either there was a base that has been deleted out of the human and orangutan sequences, or maybe the monkey and the rabbit had an insertion. So you don't know at this point if this is considered a deletion or insertion. It depends which way around you're looking at it, but it is another difference. Now, if you were to go on and look at all of this, you would notice most likely that the rabbit's going to have the most differences, the monkey the next most to humans, and the orangutan's probably going to have the least number of differences to humans. And that would give you an idea that the orangutan from that table is probably the most closely related to humans, then the monkey, and then the rabbit. The diagram on the right is what you might do with information like this. You might turn it into a phylogenetic tree. However, that diagram on the right does not use the same species as what is in the table on the left. So just be aware of that. But you can see you can take information and you can turn it into a tree like that to show the human and the chimp are going to be the most closely related in that tree, then the human, chimp and gorilla, and then the orangutan is sort of the most distantly related out of those 4. And when you look at those genome sequences or gene sequences that they've got there in coloured with blue and grey, you can see that the human and the chimps are almost identical in terms of where the sequences are aligned and where there's a gap. And that. But then the humans and the orangutans is very, very different in contrast. Here we have exam question 3. It says DNA sequencing is often performed to help produce phylogenetic trees or to classify organisms. Sections of nuclear DNA from similar organisms are sequenced and compared for similarities. Name and describe another DNA method that is used to determine how closely related 2 species are. It's 2 marks and you need to give the name and a brief description. So you can pause the video, have a think, write down your answer, and then check it against the video linked in the description box below. This brings us now to exam question 4. It says 𝕃𝕚𝕟𝕂𝕬𝕡𝕟 Island is a small island covered by tropical rainforests in Central America. 74 different species of bats live in the forest. Bats are nocturnal flying mammals. To find their way around in darkness, many bat species emit high-frequency sound pulses that bounce off obstacles and prey. These pulses enable them to judge the distance to an object. This behavior is called echolocation. 3 of the 𝕃𝕚𝕟𝕂𝕬𝕡𝕟 Island species are described in the table on the right. The biologists wanted to establish the order in which each species had evolved from the common ancestor. DNA hybridization between the various species was carried out. After analysing the results, the scientists drew the following phylogenetic tree. A. What results would have been obtained from the DNA hybridization that led the biologists to construct this phylogeny? So you can pause the video, write down your answer, and then check it against the video linked in the description box below. Here we have exam question 5. It says samples of DNA were taken from 4 individuals. The samples went through a series of steps and the resulting DNA is shown. Using the information from the diagram, which conclusions could be drawn? A. Samples 1 and 2 are from individuals belonging to the same species. B. Treatment at step X would have included cooling each of the original samples. C. Treatment of mixed samples at step Y would have included the addition of DNA helicase. Or D. Individuals belonging to samples 1 and 3 are more closely related than individuals belonging to samples 3 and 4. You can pause the video here, write down your answer, and then check it against the video linked in the description box below. So, like I mentioned earlier, instead of just comparing individual sequences, you can do whole genome comparison. This has a very creative name of comparative genomics, and that's the field where you would be doing this in. As with all our other techniques, the more connections, the more matches you have with the sequences or with the genetic material, the more similar the organisms and more closely related they are considered to be. In this case, we'll be talking about percentages, so a high percentage value indicates more similar organisms compared to a low percentage one. So here our example has human DNA being compared with the DNA from 12 other vertebrate species, and you can look at the DNA from those non-human species are aligned with and matched to the human DNA, and that's what's put in the graph to the side there. You've got 4 different kinds of DNA we're talking about. It's all nuclear DNA, but we're looking at coding DNA, the bit of the nuclear DNA that actually codes for proteins that gets transcribed and translated. That's in blue. It's a very small line across all of them down the bottom. Then in pink, you have what's referred to as UTR, or untranslated regions. These are regions that might have important information in them, but they actually don't code for proteins. They might code for other things, or maybe it's like a enhancer sequence is included within that section. Then you have the green, which is repetitive. These are sequences that are repeated within the genome. They're not translated. This might be something like satellite DNA. And finally, you have the purple section, which is not annotated. It's DNA, but we don't know what the purpose is. It's either not specified or it's unknown. But you can compare that as well still. And you can see there that between humans and chimps, we share a significant portion of all that DNA compared to, say, humans and zebrafish, where we seem to have no or not annotated or repetitive or UTR DNA in common. We have a little bit of coding DNA, and that's about it. So you can see there's quite a number of differences there. And from here, you'd be going, clearly humans are most closely related to chimpanzees, then baboons and cats and dogs and cows and pigs, and all the way down to zebrafish, which they have some relationship to, but it is very distant. And so that's how you can compare genomes to get an idea of how related two organisms are. What we can then do is we can use all this information to help predict evolutionary relationships. And those are the sorts of things that we will represent in phylogenetic trees. Here's a little example of how we might use some of that information. Here we have sparrows, brown bears, cats, jaguars, and elephants, and we've got a DNA sequence for a specific gene, for instance. What we've then done is we've taken that and we've counted out what are the differences between each combination. So obviously a sparrow and a sparrow is going to be exactly the same, so we've not included that. And if you do sparrow and brown bear, it's the same thing as doing brown bear and sparrow, so we've only done that once. That's what we've done in the table below. And what we've done is gone through and we've gone, okay, sparrow compared to brown bear, how many differences are between them? There's 8. Sparrow to cat, again, there's 8 differences between them. Sparrow to jaguar, 8 differences. Sparrow to elephant, 7 differences. Brown bear to cat, 4 differences. Brown bear to jaguar, 4 differences, and so on and so forth till we have that table filled in. From there, we can actually then take that information and we can turn it into a phylogenetic tree, where we go, who's the most closely related? The cat and the jaguar, so they're going to be the most recently branched away from each other. Then we'd look and say, well, the brown bear and the elephant are pretty close, so they're going to probably also have their own branch. And then we'd probably join those two groups together. When we're looking at the relationships here, we can see that those two groups sort of are about 4 away from each other, 4 different from each other in both sets. And then finally, you probably have the sparrow as a separate thing on that tree because it is most distantly related to the others. And so from there, we can take information like this, which can be a little tricky if you've just got a whole sequence of letters and how do I interpret that? And it's very easy to get lost in it, even so with a table like this. But you can take that and take a phylogenetic tree, which is a really handy way of visually representing this information and quickly communicating the key points about who is most closely related and who is most distantly related. And so this is a sort of technique that we might use to do that. Stepping now away from looking at DNA, we can compare amino acids. And we can do this because all organisms are using the same 20 amino acids to make their proteins. But again, the same rule applies. If we look at the sequence of amino acids for a protein, and there's lots and lots of differences, they're going to be less related. If we have low differences and lots of matches, they're going to be more related. And so the idea is here is still the same. You compare the sequence of amino acids, look for how many similarities, how many differences. The more similarities they have, the more related they are. The more differences they have, the more unrelated they are, or more distantly related they are. The trick here is, again, just like with DNA, when you are comparing DNA, you have to look at similar or the same genes. When you're comparing amino acid sequences, you need to be looking at the same proteins. And so here you have to be careful and you have to be able to find a protein that is present in all the organisms. So one place to look for proteins that are present in all organisms is to look at processes that occur in all organisms. Things like protein synthesis, DNA storage, ATP production. These are things that most organisms and most cells do to some extent. So any of the proteins involved in them are going to be really good candidates for this process. Organisms that have blood often are going to use hemoglobin as part of forming those blood cells. And so we can look at the chains of hemoglobin to work out how similar or how different they are. And that's what the diagram on the right is showing there. 𝗍𝗂𝗃 is another molecule that we could look at as an example. It's an enzyme involved in ATP production in the mitochondria. Because lots of organisms are going to be using mitochondria to produce ATP, it's pretty easy to use 𝗍𝗂𝗃 because it's going to be a consistent molecule in all of those situations. All organisms in the 5 kingdoms have it, so it's widespread. It's a great example of something we can use for amino acid comparison. Now here in our table below, we took the amino acid sequence for 8 different species, looked at their 𝗍𝗂𝗃 sequence and compared that to the human 𝗍𝗂𝗃 sequence. And we've highlighted in red where it is different from the human sequence. If it's in green, it's the same. One thing to note about the table at the bottom is that amino acids have 3 ways that you can write out their name. The first way would be to write out the full name, something like lysine. The second way would be to use the 3-letter code, so lysine might be LYS as its 3-letter code. And the third way is a single letter code that you can use, and that's what they've used here. And so you can see that's why we have like F and G and R, K, T, Q, A, P. These are representing different types of amino acids. It's just a 1-letter code. A little bit like how the periodic table has a 1 or 2-letter code to represent different elements, this is a 1-letter code to represent the different amino acids. And so you can see it's all written out here, and that, for example, chimps have no differences to humans. They're completely the same, so they're obviously going to be quite closely related. The rhesus monkey has one difference, which would mean it's probably a little bit less related or less closely related to humans than the chimpanzee is, whereas like the tuna fish and the lamprey have 20 and 21 differences between them compared to humans, so they're going to be quite more distantly related than a chimp is going to be to a human. And so that's how we can look at these sorts of information and we can work out relatedness from them. So you might be wondering at this point, why bother with these 2 different techniques? Wouldn't DNA be the best all the time? Why would we bother with amino acid? DNA is going to be really, really precise. Well, it comes down to a range of different factors. You might not have the money or the equipment to do one or the other, but it also comes down to how related you think they are going to be. So if you think you've got species that are closely related, then you're really going to want to look at their genetic differences because they might not show it with their amino acid sequences. If you've got 2 species that you think are really distantly related, it might be easier to look at their amino acids and see if where the differences are there, because that might take a little bit longer for them to accumulate. It might be a bit much if you look straight at their DNA. You might also find that you don't have the equipment to do DNA, so you do amino acid over that. So there's a couple of reasons that you might pick one over the other. Now, if you have an idea of how many differences there are between 2 organisms and how quickly those organisms occur, you could work out how long ago those 2 organisms diverged. And this idea of knowing how frequently differences accumulate is this idea of a molecular clock. So genes and protein sequences will accumulate mutations at what we call a relatively constant rate. So you might see 1 change per million years, or it might be different. And so you use this like a little clock. The number of changes you see to multiply by the number of years it takes for 1 change to happen, and that's going to give you the number of years it's been since those 2 species diverged. And so you can see the example there. If you have a molecular clock rate of 1 base pair change per 100,000 years, then any species where you might have 6 base pairs being different, that would be 6 times 100,000, telling you that they must have diverged 600,000 years ago. So this is a really handy thing you can use to calculate backwards how long ago 2 species may have diverged from each other. There are some limiting factors here, however. 1, not all genes and proteins actually change at the same rate. So hemoglobin might mutate more rapidly than 𝗍𝗂𝗃, so you can't use the molecular clock for one protein or gene for another one necessarily. The other thing that's a problem is that between different species or different groups of organisms, you may see faster or slower rates of change. So the clocks could vary between them as well, and you'd have to have some way of accommodating that. And the other issue is a bit of an odd one, but it essentially goes like this, that the more time you have that has passed, the more chances there are for one of those early mutations to become reversed by a later mutation and essentially become hidden. Instead of looking like 2 mutations have occurred, it looks like no mutations have occurred at that point. And so it can actually make it look like less mutations have occurred, therefore making your overall analysis less accurate. Because instead of thinking you've had 6 mutations, you might be thinking you've only had 4 mutations, but in reality, it's a bit longer. So here we have exam question 6. It says chimpanzees are the closest living relatives of humans. Less than 1% of our DNA has changed in the 6 million years or so since the humans and chimpanzees lineages diverged. The measurement of the rate of accumulation of random genetic changes in the 2 genomes of chimpanzees and humans is called A, stratigraphy, B, a molecular clock, C, radiometric dating, or D, relative dating. So you can pause the video now, write down your answer, and check it against the video linked in the description box below. Let's now talk about how you might represent evolutionary relationships. We've touched on this briefly before about using phylogenetic trees, but we're going to dig into them a bit more detail now. So we can use these trees as a graphical way of representing these relationships. They're similar to family trees in a sense, and instead of showing sort of more recent family ancestry, they're showing an evolutionary ancestry. And so those relationships, divergence, any common ancestors, those should all be represented here between different species. The structure looks like this. The root, you can see, is on the left there. That is your line at the origin. This is your earliest common ancestor that you have for all the species in this phylogenetic tree. Coming off that, you will have branches. These are just the lines on the phylogenetic tree. So all lines here are branches. Any point where you have branches split and become more branches, these are nodes. And this is representing a divergence between 2 taxa, 2 groups of organisms. An external node is sometimes also called a leaf. This is one towards the end. This is representing the present day organisms or any extinct taxa or species. So if you've had something die off, and it will become extinct, this would still be considered an external node. It's sort of the most recent end of that branch. A clade is a bigger group where you're talking about a whole big group of organisms that have a particular ancestor in common. And then taxon is just the term we use to mean it's a scientifically classified group like a species. So that is sort of all the different components of your phylogenetic tree. How then do you read them? Well, first find the species that are listed. These are really your present day species. Occasionally you might have something noted as being extinct, but they're generally considered the most modern species in the tree. Going back along the lines will take you back in time towards the ancestral species until you get to the root, which is the earliest ancestor of all those present day species that are listed. As you're going back, you'll come across branching points or divergences, and this is where you've had an ancestral species that has split and formed 2 new descendant groups. And so at each of those branching points is what we would call a common ancestor. And depending on who comes from that down the line, you would say, for instance, the top one there is the most recent common ancestor of just A and B. But if you go back earlier, it's the most recent common ancestor of A, B, C, D, and E. So it's all of them. And so you have to go back quite a few points to find that one. There are a few steps when it comes to making a phylogenetic tree. The first one is you're going to need to know how many differences are there between all the different species or groups involved. Then you're going to go who has the least differences. Those are the 2 that will be most recently diverged from each other. Then you're going to kind of keep working through that. Once you've made that group, you're going to find the next group has the next fewest differences, join them with a node. Again, find the next one with the fewest differences and add that to the tree as a branch. And you're going to continue that until all the groups are added onto this phylogenetic tree. If there's an outgroup, add that. What's the most distantly related one? Place that on the tree. You can see here on the right, we've got sort of more of an angled phylogenetic tree. It's the same thing. At the top here are the modern day species, and if you go down the diagram, that's where you're going back in time. But it's still the same idea. The one at the top is sort of the flipped version of that, with the present day species are at the bottom, and then going up the diagram is taking you to the common ancestor. So the top diagram and the bottom diagram are sort of inverted versions of each other almost. So the top one you read from sort of bottom to top, and the bottom one you read from top to bottom to go back in time. So this video is going to take you through a detailed way of how you can do a phylogenetic tree with some data that might be provided to you.
Chapter 4. Building phylogenetic trees. A phylogenetic tree is a diagram showing inferred evolutionary relationships between a set of organisms. Phylogenetic trees have a branching pattern, like this figure from Darwin's notebook, where he has sketched out this branching pattern to reflect the process of descent with modification, a process central to Darwin's theories of evolution. This second tree shows the inferred evolutionary relationships among Darwin's finches on the 𝗃𝕚𝕚𝕚 Islands. But how are phylogenetic trees like this one built? Well, any characteristic can be used to infer relationships and build phylogenetic trees, and DNA sequence data has proven incredibly helpful in clarifying and building many important phylogenetic trees. Using large DNA datasets to build trees can get quite complicated, but the underlying principle and logic is pretty straightforward. Sequences separated by shorter evolutionary distance are expected to be more similar to one another than sequences separated over longer evolutionary distances. We'll go through a highly simplified example to familiarize ourselves with the basic logic of phylogenetic tree building from DNA sequence data. We will do this in a series of steps so that you can pause the video at any point to work through the steps yourself, returning to the video to check your progress. In our simplified example, we have a set of 6 sequences. These are the equivalent stretch of sequence taken from the equivalent gene from each of the 6 species, and we're just going to refer to the species as A through F. The first step in the process is to line the sequences up against one another. Here we have the sequences aligned so that the equivalent nucleotide in each sequence lines up to form a column. This makes comparing the sequences much easier. With the sequences aligned, we can now compare each sequence with one another. We'll do this in a pairwise fashion, starting with sequences A and B. So our next step is to determine how many differences there are between sequence A and sequence B. When we do this, we see that in 3 positions the sequences are the same, and in 9 positions they are different. So we have 9 differences between sequences A and B. We can start building a table of all the differences between the sequences, and our first entry is 9 differences between A and B. The next step is to compare sequences A and C to determine how many differences there are between these 2 sequences. In this case, we can see that the sequences are much more alike, with 10 positions that are the same between the sequences, and only 2 that are different. So our next entry in the table is 2 differences between A and C. And we can continue comparing the sequences in this way until we have completed the rest of the table. This is our next step. And this is what the completed table looks like. With the table completed, we can move to the next step, which is to use the table to identify the sequences with the fewest differences between them. We will infer that these are the sequences that are the most closely related to one another. In our table, we see that the sequences with the fewest differences between them are A and C, with only 2 differences, as well as B and E that also only have 2 differences between them. With this information, we can draw the first groupings on our phylogenetic tree. We'll group A and C together to reflect the fact that these 2 sequences show the closest relationship we have here to one another, and we'll group B and E together to reflect the fact that they also show an equivalently close relationship to one another. With the first 2 groupings made on our tree, we now need to rework our table, with the grouped sequences combined together as a grouping rather than 2 individual entries in the table. We'll start by combining A and C, this group. To do this, we'll take the average difference that A and C show to each of the other sequences. Let's start with the differences they show to B. We see there are 9 differences between A and B, and 9 differences between C and B. So the average difference of A and C to B is 9, and we can make that entry on our new table. Move to the next position, D. We can see there are 4 differences between A and D, and there are 5 differences between C and D. So the average difference of A and C to D is 4.5, and we can add that entry on our new table. We can complete the rest of the table in this way. We have an average of 9 differences between A and C to E, and 10 differences to F. The rest of the table can be copied down from the first table. With the AC grouping now added to our new table, we can proceed to also add the BE grouping. And to complete the table with B and E grouped together using the same approach as before. For B and E, we have an average of 9 differences to the AC group. We have 4.5 for AC to D and 10 for AC to F. For B and E to D, we have an average of 6 differences and an average of 10 for B and E to F. And there are also 10 differences between D and F. With this table completed, we can now proceed to the next step. This step is to identify the sequences with the fewest differences between them in our new table. We can see that the AC group has only 4.5 differences to D, so this is the next close relationship in our tree. And we can add this next grouping to our tree, like this, with D as the next grouping out from A and C, reflecting the fact that D is more closely related to them than it is to the other sequences we have. With this new grouping added to the tree, we need to rework our table again, with the ACD grouping incorporated. We have an average of 7.5 for AC and D to the BE group, and we have an average of 10 for AC and D to F. And we also have 10 differences between the BE group and F. With the new table completed, we can now determine the next relationship by identifying the sequences in the table with the fewest differences between them again. We can see that this is the ACD group with the BE group, with an average of 7.5 differences. So we can add the next grouping to the tree, like this, grouping the ACD group with the BE group. This now leaves us with one more sequence, F, that is equally distantly related to all the other sequences, with 10 differences to each of them. So we can add this last grouping to the tree like this, reflecting the distant relationship between F and all the other sequences. And this completes our phylogenetic tree built from these 6 DNA sequences. The method we use to build this tree is a distance method, specifically an approach called unweighted pair group method with arithmetic mean, or UPGMA. And the approach we took works well for a straightforward situation like this, but when we start to look at larger and more complex data sets of DNA sequences, things can become more complicated, and we need to start taking a number of other factors into account. And it's often a good idea to compare the results from multiple different approaches to develop a sense of how reliable different parts of your tree are. But despite these potential complications, the underlying logic remains that sequences separated by shorter evolutionary distance will be more similar to one another than sequences separated over longer evolutionary distances. DNA sequences therefore provide a powerful source of information to help us infer evolutionary relationships that can be depicted in the form of phylogenetic trees.
Here now we have exam question 7. It says 2 possible phylogenetic relationships between 8 groups of flowering plants are shown in the following diagrams. One similarity between the alternatives is A. Monocots diverge before chloranthales, B. Ceratophyllum and eudicots diverged from monocots, C. Amborella and Nymphalis diverged first from Angiospermae, or D. Magnolids were the first group to diverge from Mesangiospermae. So you can pause the video, write down your answer, and check it against the video linked in the description box below. When we build a phylogenetic tree, it is possible sometimes to come up with more than one result. That can come about because of a range of different things. One, you might not know the exact divergence point. Maybe the fossil evidence you've got isn't super accurate. Maybe there's a couple of ways of interpreting data. And so here, the way you might express that, you can see here is, for example, with Y and Z, there's a bit of a gap there because you don't exactly know where that divergence point is. You just know it should occur somewhere around there. Or for W and X, W might be an ancestor of X, but we don't see that transitional fossil, so you have a break between them. There's a little bit of a gap. For species S, the line is really short there because it became extinct, so it doesn't reach the end with all the other letters. You might also have, for instance, where T, U, and V are, there might be a little lack of clarity who diverged from who here. Maybe you weren't just looking at fossil evidence, maybe you looked at genetic evidence, but the similarities are all really close, so it's not clear who's more closely related than who. Maybe you need more data for that. Or maybe the speciation event occurred at really similar times for all of these species, and so they're all coming out of one point there. So sometimes there are things with phylogenetic trees we just don't know, and we have to include those uncertainties within it. It's also possible that you can have crossbreeding occur in phylogenetic trees. And so even after groups have diverged, sometimes genetic material gets passed back and forwards. Particularly around human evolutionary history, there seems to be strong evidence suggesting that certain groups of modern humans, Homo sapiens, did interbreed with Neanderthals, also known as Homo neanderthalensis, which caused part of their genomes to be passed between each other. Now, this certainly causes questions as to whether or not they should be considered two different species, because we know that organisms of different species can't produce fertile offspring, and here, if they've had descendants come from these crossbreeding events, that would seem to imply that maybe they are the same species, not different species. But if you do have interbreeding events, and this could occur before a point where full speciation has occurred, you draw that with a line across those branches to indicate an interbreeding event. So you can see here we've got two diagrams, and they're both really representing the same information. They're just doing it in different ways. And so you can have a look at those and how they've gone about doing that. You've seen a couple of different types of phylogenetic trees, but phylogenetic trees can look a number of different ways. They still have the same fundamental structure, but depending on what sort of shape you make that tape, you'll end up getting different sort of looks to those phylogenetic trees. And it really comes down to what angle are the lines on, and are you using straight or curved lines or some combination thereof. You will also find that some phylogenetic trees may not actually appear to have a clear root ancestor. That's fine, that's done intentionally because they don't know of one. Or it might also be because that the organisms may have evolved through adaptive radiation, so it's very unclear where that sort of starting point is because everything sort of came about at the same time. So you can see here we've got a few different kinds. In the bottom on the left, you've got the sort of the more classic shape that we've been looking at, and that's a very similar one to the bottom on the right in the set there. It's just rotated vertically, but you also have a V-shaped one. We've seen those as well before. And then in the middle you have a more curved one, and if you were to take that curved one to the extreme, you get the diagram that is on the right there with all the dogs and that, and all the different breeds that they've got and how they've diverged. And this is a very radiated one, so that's why it's represented like this. When we look at phylogenetic trees, we also have a few different ways we can group the organisms there. And there's three main ways that we're interested in. We're looking at monophyletic, polyphyletic, and paraphyletic. And this is sort of really considering where is their common ancestor in these groups. So a monophyletic group is the one in red on our diagram there. This is where the group of organisms you're referring to includes a common ancestor and all its descendants. So you can see there the common ancestor C is selected, and so is all of the descendants from C. Polyphyletic is where you have a group, but it's not including the common ancestor. So here, it's the blue one in the diagram. The common ancestor for all those organisms that have been selected would be D, but it's not actually included in this. So you've actually got lots of little monophyletic groups together, creating a polyphyletic group. Paraphyletic is an interesting one. You have the common ancestor, but you maybe didn't include all of the descendants, and that's the one in green on the diagram. So you can see that B was included, and most of B's descendants were as well, but not H and its descendants, even though technically they are descendants of B. So this is paraphyletic, where it's most of the phylum, most of these organisms, but not all of them, some that have been excluded. So those are the three ways we can group organisms. There's also a technique known as cladistics, which is where we classify organisms as the groups of species that we would otherwise call clades. And this is very imaginative because it comes from the Greek word kleidos, which means branch, because we're grouping branches on the phylogenetic tree. So that's where the word clade comes from. Now, the description of a clade is it's an ancestral organism and all its evolutionary descendants, which might sound familiar because we just established that as the definition for a monophyletic group. And so monophyletic groups are clades as well. They're actually the same thing. And so what you find is that members of one clade will have common characteristics because they have that shared evolutionary lineage. You can see there that the blue and green examples are clades because they're an ancestral species and then all the descendants, whereas the pink and orange might have some ancestral species, but they're not covering all of the descendants from that. The yellow one would be more considered to be a paraphyletic group where you have the common ancestor and then not all the descendants, whereas the red one is more of a polyphyletic group where you have a couple of actual monophyletic groups sort of in there, but not all the common ancestors included. This leads to two diagrams that look very, very similar but are slightly different. Phylogenetic trees, or phylograms, and cladograms. For the most part, they basically represent the same information across the board. But there is some key differences. In a phylogenetic tree, technically, you would vary the branch length based on the amount of time. So branch length means something. In a cladogram, branch length doesn't mean anything. It's all the same across the board. And so phylogenetic trees, if you are drawing branch lengths or branches of different lengths to represent more or less time since divergence, that's what a phylogenetic tree is, and you're going to do that so that you could infer the amount of evolutionary time that separates two species. Cladograms, on the other hand, because they don't care about the length of those branches and they're not anything to do with how much time has passed since speciation, you can't infer the amount of evolutionary time that separates two species from a cladogram. But they are representing the splitting of all those different common ancestors into the new modern groups. They are representing the same information, just phylogenetic trees will be able to have time since divergence inferred from them, and cladograms will not. Exam question 8 says part of the phylogenetic tree for bats is shown on the right. The most recent divergence of two bat species is the A, smoky and fishing bats, B, evening and long-fingered bats, C, slit-faced and sheath-tailed bats, or D, sucker-footed and New Zealand short-tailed bats. So you can pause the video, have a think and write down your answer, and then check it against the video linked in the description box below. This brings us to exam question 9. In humans, severe acute respiratory syndrome, or SARS, is a serious form of pneumonia. SARS is caused by a coronavirus that was first identified in 2003. Scientists suspected that the virus had been transmitted to humans from some other animal. Testing was completed on several animal species. Strains of the coronavirus similar to those found in humans were identified in different species of horseshoe bats, genus Rhinolophus, and palm civets, 𝕃𝕚𝕟𝕠𝕟 levata. Samples were taken from the different sources, and the virus's RNA from each sample was sequenced. What molecular information were the scientists obtained from sequencing RNA? The molecular information enabled the scientists to draw an evolutionary tree for different strains of the coronavirus. The evolutionary tree on the right was drawn. Coronavirus strains 2 and 3 are the most similar of the strains. B. Using your knowledge and the information given in the evolutionary tree, give 2 possible explanations as to why they are the most similar of the strains. Strain 7 is found in palm civets, and strains 5 and 6 in humans. All other strains are found in different species of horseshoe bats. C. What conclusion can be drawn about the origin of the strain of virus that causes SARS in humans? So pause your video, write down your answers for A, B, and C, and then check them against the video linked in the description box below. So here we come to an activity that you'll have a chance to do in class, but it is also the end of this PowerPoint. If there's anything you didn't understand, you can go back and rewatch that section or reach out to your teacher for further clarification. Otherwise, I'll see you in the next video. Bye!